So for this particular project, we wanted to test as to whether dung beetles can be used as sources of environmental DNA to detect the diversity of vertebrates within tropical rainforests, and also as to see if we can establish those direct interactions between individual dung beetles as well as the kinds of vertebrate and mammal dung that they are fed on. So to do so, we did a survey within the Central Catchment Nature Reserve in Singapore. We actually set up traps across 50 sites across this nature reserve. So after 24 hours, we collected these live dung beetles and immediately we preserved them in 100% molecular grade ethanol to preserve their DNA. So after dissecting out the gut contents of every individual dung beetle and extracting the DNA from those gut contents, the next step is to amplify whatever kinds of vertebrate DNA that can be found in those samples. So to amplify specifically vertebrate DNA, what we use two different types of primers. The first primer is also known as the 16S rRNA primer, and then we use another set of primers also known as the 12S rRNA. From this particular field survey, we were successful in sequencing different kinds of vertebrate DNA from the individual dung beetles that we collected. From over 500 individual dung beetles, we actually were able to detect 18 different species or native vertebrates that are found in Singapore. So this was actually really interesting because we managed to detect the DNA of rare and elusive mammals. This included the endangered Sunda slow loris as well as the Sunda pangolin. That information, we were then able to construct the ecological networks that show these direct interactions between the dung beetle species and the vertebrate species in each of those 50 sites that we did our surveys across the Central Catchment Nature Reserve. And when we looked at these networks in more detail, we then found that these networks were actually pretty similar all across the Central Catchment Nature Reserve. So there wasn't really any difference in the structure of these networks across that space. But having said that, the fact that we were able to construct so many networks across this wide spatial area, the fact that we're able to demonstrate this really shows that dung beetles can indeed be used as an effective resource of environmental DNA, and then we could use that to construct these ecological networks.